Good morning everybody. I welcome you all to this video lecture Light Phase. The video lecture is being presented by D. Rajashekhar, lecturer in Botany, Srimati Ganganeni Kalyani Government Degree College, Vinikonda, Guntur district. Before entering into the real lecture, actually I would like to recall some of the important and basic details of photosynthesis. Photosynthesis is basically an anabolic pathway or constructive process where different simple molecules come closer and they enter into reactions so that they form a complex substance. By this process, oxygen is also liberated as a byproduct. Plants prepare carbohydrates by this photosynthesis process. Carbon dioxide is taken and oxygen is liberated. Actually, photosynthesis is broadly classified into two different phases. They are light phase and dark phase. As we all know, chloroplast is the major site of photosynthesis where thylakoids, which are also called together as grana region, where light phase takes place, and the stroma region of chloroplast dark phase actually takes place. What is light phase and what is dark phase? So basically photosynthesis is energy changes. During light phase, the solar energy is changed into chemical energy, nothing but ATP and NADPH2. They are also called assimilation power, and thus released chemical energy is utilized in dark phase so that potential energy is formed, that is carbohydrates. Actually, light phase can also be done in two different processes. They are cyclic photophosphorylation and non-cyclic photophosphorylation. Photophosphorylation means formation of ATP in the presence of light. It can be done either by cyclic method or by non-cyclic method. In cyclic method of photophosphorylation, we can expect only ATP. Two ATP are released at two different places during the electron transport system. During electrons are getting transported, a type of force is developed that is called proton motive force. It is actually formed because of the proton gradient, and this proton motive force helps in the formation of ATP. So electron transport system or chain indirectly involves in the formation of ATP, but the direct involvement in the formation of ATP is because of proton gradient and proton motive force. All these things can be discussed at the end of this video lecture. So in this slide you can see cyclic type of photophosphorylation. Actually it is a cyclic process. It is basically electron transport system where different electron carriers and photosystem 1 are getting involved. So if you see clearly, the photosystem 1 with reaction center P700 is the place where electrons first generate. From there, these electrons are carried through different electron carriers and again they reach to the primary site, that is a P700 or first reaction center. As all these reactions of electron transportation appear like a cycle, this is called cyclic photophosphorylation. Actually, cyclic photophosphorylation is seen in higher plants as well as in bacteria, lower organisms. First and foremost, we have to know a little about photosystem 1. As we all know, photosystem 1 has two parts. One is reaction center, that is P700, and the antenna, together called light harvesting complex. First and foremost, these antenna molecules, nothing but chlorophyll molecules, where the reaction center is only single chlorophyll A molecule. Taking solar energy, they get activated because of this solar energy. That solar energy is carried to other chlorophyll molecules present in antenna by inductive resonance process. Finally, that energy is given to reaction center, that is P700. Because of activation of this reaction center, it moves to the excited state, and within 10 to the power of minus 9 seconds again it comes back to its original state. So then the observed energy from the sun is liberated in the form of electrons. Thus two photons are changed into two electrons. These two electrons are carried through ferredoxin, plastoquinone, cytochrome B6, cytochrome F, plastocyanin, and finally to the same place, that is photosystem 1. When these reactions are taking place, at two sites ATP are generated. How these ATP are generated will be discussed in the end of this video lecture. Thus, in cyclic photophosphorylation, the reactions are cyclic, and here different electron carriers are getting involved. Only photosystem 1 is involved in this process. Only ATP are produced. The total amount of ATP we can expect out of the cyclic photophosphorylation is just 2. And then non-cyclic photophosphorylation. Non-cyclic photophosphorylation is generally seen in higher organisms. Actually here the reactions are not cyclic, hence it is named as non-cyclic photophosphorylation. Here also ATP are generated. Along with ATP, NADPH2 are also produced. Here two photosystems are involved in non-cyclic photophosphorylation. As it seems like Z, letter Z, it is also called Z scheme. Here actually the process starts with the photosystem 2. Photosystem 2 is with the P680 reaction center. Just like cyclic photophosphorylation, here also photosystem 2 receives solar energy in the form of photons, so that the reaction center is excited, and within a short span of time it comes down, so that the photons are changed into electrons, which are transported through plastoquinone, cytochrome B6, cytochrome F, plastocyanin, from plastocyanin to photosystem 1. So that photosystem 1 is also excited, and the two electrons are given to ferredoxin reducing system, from there to ferredoxin, and finally these two electrons reach to NADP molecule. For continuous operation of photosystem 1, electrons are being supplied by photosystem 2. For continuous operation of photosystem 2, the electrons are supplied by water molecules. Water molecules undergo photolysis, that is, in the presence of light, water molecules get broken down into electrons, protons, and oxygen. The electrons are given to photosystem 2 for continuous operation of it, and oxygen is liberated out, and hydrogens are given to NADP, so that NADP is reduced to NADPH2 in the presence of an enzyme NADP reductase. NADP reductase. Thus, actually, the non-cyclic photophosphorylation occurs. Here in this type of photophosphorylation, you can see the formation of ATP as well as NADPH2. Here, along with the light harvesting complex, photolysis of water is also happened, so that both photosystem 2 and 1 are put in continuous operation. The reactions occur in a non-cyclic fashion. So while electrons are getting transported from one system to another system, and similarly from one electron carrier to another electron carrier, what happens actually? We all think that electrons, as they get transported, as we have seen, ATP are generated, but it's not true or real. Actually, electron transport system is a force by which proton gradient takes place. What is proton gradient? Difference between the proton ion concentration on both sides of the membrane. As we all know, light phase takes place in the thylakoid membranes where photosystem 1 and 2 are present. Here in this slide you can see, there are four complexes. They are photosystem 2, cytochrome system, photosystem 1 and ATP synthase. They are shown in different colors. All these four complexes are tightly embedded in the thylakoid membrane. Outside the membrane you can see chloroplast it is clearly written there, and inside the thylakoid membrane you can see thylakoid lumen. When electrons are transported from photosystem 2 to cytochrome system or complex, in between them there is a molecule called pheophytin. First of all, photosystem 2 gives 2 electrons to this pheophytin. It is also a chlorophyll molecule, like a chlorophyll molecule, but the difference between the chlorophyll molecule and this pheophytin is, in pheophytin magnesium ions are absent in the center. So it's like a chlorophyll molecule. This pheophytin, receives 2 electrons from photosystem 2 as well as 2 protons from the chloroplast stroma, so that this quinone is changed into plastoquinol, that is PQH2. The so-called conversion of plastoquinone and plastoquinol is called quinone cycle, which operates between the photosystem 2 and the cytochrome complex. So the received protons from chloroplast stroma by this quinone cycle, again these electrons get entered into the thylakoid lumen by the same quinone cycle. Thus, the hydrogen ion concentration is decreased in chloroplast stroma region and it is increased in the thylakoid lumen. So there is a difference in the concentrations of hydrogen molecules or ions present in the two regions which are adjacent to this membrane. So because of this proton gradient, a force is developed that is called proton motive force. That is nothing but the balancing the hydrogen ion concentration on both sides. Because of this proton motive force, the hydrogens which are present in high concentration in thylakoid lumen, again they will be taken back to the chloroplast stroma through ATP synthase enzyme. Here in this slide you can see orange color region, that is actually ATP synthase enzyme. This ATP synthase enzyme consists of two parts. One is CFO, coupling factor 0, and coupling factor 1. Coupling factor 0 is also called a tail region, which is firmly embedded in the thylakoid membrane, whereas the head region, that is coupling factor 1, is freely revolving in the chloroplast stroma. Thus, when hydrogens are passed through quinone cycle from chloroplast stroma to thylakoid lumen, these hydrogens are again taken back to chloroplast stroma through ATP synthase enzyme. When 4 hydrogen ions enter from thylakoid lumen to chloroplast stroma, 1 ATP is released. Thus, the ATP are synthesized by this proton motive force, which is developed because of proton gradient across the membrane. That is called photophosphorylation. It is mediated by the electron transport system and electron carriers. So, in non-cyclic photophosphorylation, NADPH2, 2 NADPH2 are formed actually, and 1 ATP is formed. So when we see the differences between the cyclic photophosphorylation and non-cyclic photophosphorylation, in cyclic photophosphorylation, only photosystem 1 involves, whereas in non-cyclic photophosphorylation, both the photosystem 1 and photosystem 2 get involved. In cyclic photophosphorylation, as photosystem 1 only involves, the reaction center of it is always P700. It receives always higher wavelength of light from the sun. Whereas in non-cyclic photophosphorylation, the reactions start with photosystem 2, whose reaction center is P680 actually. In cyclic photophosphorylation, electrons are released they run the entire cycle and they come back to the reaction center again. So that reactions are all in cyclic manner. Whereas in non-cyclic, the electrons are not revolved in a cyclic fashion, but in non-cyclic fashion or in the form of a letter Z. Hence the name Z scheme. Here in cyclic photophosphorylation, there is no photolysis of water. Water molecules do not get broken down and they do not give oxygen and electrons and protons. But here in non-cyclic photophosphorylation, the hydrolysis of water, that is also called Hill reaction, takes place so that oxygen is evolved, protons are released and electrons are released. In cyclic photophosphorylation, only 2 ATP are released at 2 different points. Hence, that is called substrate level phosphorylation or photophosphorylation. Here, in the case of non-cyclic, 1 ATP is formed at one place and 2 NADPH2 are formed actually. In cyclic photophosphorylation, there is no requirement of external electron donor. Actually, photosystem 1 is activated by the solar energy that is taken in the form of photons actually. But here in non-cyclic photophosphorylation, for the continuous operation of photosystem 2 and indirectly photosystem 1, electrons must be supplied. That is done by the photolysis of water in case of higher plants and H2S in the case of bacteria. There is a herbicide. It is also an algicide, that is dichlorodimethylurea. It does not involve in cyclic photophosphorylation process, so that it is continuously operated. But in case of non-cyclic photophosphorylation, the same DCMU, that is dichlorodimethylurea, it obstructs the electron passage from photosystem 2 to plastoquinone. That is why when the electron passage is stopped automatically, the non-cyclic photophosphorylation is stopped, and abruptly it is stopped. When it is restored from the effect of DCMU, then only it gets operated. That is why non-cyclic photophosphorylation is sensitive to TCMU activity and which inhibits the electron flow. Actually, the electron transportation pattern was put forward by Hill and Bendall in the year 1960. How actually electrons get transported from one electron carrier to another electron carrier and from one system to another system. Along with this electron transport system, these ATP are synthesized. That is called photophosphorylation, formation of ATP in the presence of light. So, what we have learned from this video lecture, actually, photosynthesis is a constructive and an anabolic pathway where simple molecules are changed into a complex substance. CO2 is taken in this process and oxygen is liberated as byproduct. It is broadly classified into two phases, light and dark phases. Light phase takes place in the grana region of chloroplastid. The dark phase is generally carried out in the stroma region of the chloroplastid. Light phase is nothing but conversion of solar energy into chemical energy, that is ATP and NADPH2 formation. And then dark phase is conversion of this chemical energy into the potential energy, that is carbohydrates. This is a great process that is seen only in the green plants and some bacteria. By this process, plants prepare food material and they can be served as the primary producers for the entire globe. And also it liberates oxygen, which is an important and life retaining process for all other organisms. In the light phase, actually, it is conversion of solar energy into ATP and NADPH2. It can be done by two methods, in two patterns. One is a cyclic type of photophosphorylation, another one is non-cyclic type of photophosphorylation. Actually, ATP are not directly synthesized by the electron transport chain. By this electron transport chain, proton gradient, proton motive forces are developed by which the ATP are synthesized directly and also in the form of NADPH2. These ATP and NADPH2 are utilized in the dark phase. And we have already seen the differences between the cyclic and non-cyclic photophosphorylations. Thank you for your patient listening. I hope you have followed the entire lecture with great interest. We will soon meet with another video lecture. Till then, bye. Have a nice time. Thank you.